Hello, my name is Remy Ajani, and this is my gut microbiome presentation. To start off, I'm going to talk about what a microbiome is and what a gut microbiome is as well. So, a microbiome are the types and proportions of microorganisms living in a given location. A gut microbiome are the microbiomes in an animal's gut. So, the importance of a gut microbiome is to kind of, find the conditions that deal with your health, the environment, and also your diet. Health correlations that kind of have a physiological factor to it are depression, your weight, obesity, autism, Alzheimer's, things of that nature. And so our question was, will diet have an effect on the gut microbiome of small dogs? So we hypothesized that if small dogs, specifically Yorkie poos, are exposed to consume more processed food, their gut microbiome will be different from the gut microbiome of similar dogs their size who eat a more vegetarian diet. We kind of believed that since processed foods contain more chemicals and dyes than natural based foods, it will contain different bacteria compared to the natural based foods as well. So the description of our animals selected was that we had sample 333, which is a 6-year-old 8-month-old Yorkie poo. His diet contained grain-free, dry, simply nourished. And while our other sample was 342, it was a 6-year-old, 1-month-old Yorkie poo. His diet contained celery, carrots, and milk bones. So that was more of the vegetarian diet. And two of the samples that we got from another group was sample 343. It was a 3-month-old husky. His diet contained milk bone, dry food, while 344, that sample, was a 2-year and 5-month-old husky. His diet was just a raw diet. So basically, there were 3 key steps with analyzing the DNA. We had to isolate the DNA. We had to isolate the DNA, amplify the DNA, and kind of determine the nuclear sequence of the DNA. So the first thing we did in this experiment was use the Chiogin kit, which is to isolate the DNA from the stool by using a variety of buffers that prevented the enzymes from degrading DNA and washes to wash away impurities and unbind the DNA. Next, we used the Zymo kit. We used the Zymo kit and a variety of buffers to filter out the impurities from the DNA to concentrate the DNA more by changing the volume from 200 microliters to 20 microliters. After that, we used the nanodrop. We loaded samples onto the nanodrop to determine if there was any contamination from proteins or even RNA within our sample by figuring out the concentration of each sample and measure the purity, which should be a 260/280 ratio. Once we isolate the DNA, we amplify the V3 and V4 regions of the 16S ribosomal RNA gene by using PCR, in which the size after amplicon will be around 550 base pairs after 25 cycles. We used the gel electrophoresis to check the products of our PCR reaction by separating DNA fragments of different sizes. And we, for the PCR cleanup, we used the Agent Court Ampure XP kit to clean up the amplicon from our amplification PCR reaction. This kit will remove components of the buffer and unused primers by using magnetic beads as the solid surface, which will allow the double-stranded DNA of the amplicon. After that, we kind of did some indexing with the PCR. So in order to determine the nucleoid sequence of the 16S ribosomal RNA that was amplified, we ran 8 cycles of our PCR reaction that kind of was a thermal profile. So we kind of had to, anneal and degrade and kind of like heat up and stuff like that to add short sequence tags to the ends of our amplicons. We used the fluorometric assay to determine the concentration of our amplified sample after the PCR cleanup. We use this rather than a spectrometer because it's more accurate with distinguishing between DNA, RNA, and proteins. We sent our samples off to the next-gen Illumina sequencing for sequencing. So now we're going to talk about the observations that we observed during this experiment. So after centrifuging the samples, fecal sample 333 had a grayish-brown supernatant and a thick pellet, while samples 342 and 343 and 344 had a reddish-brownish supernatant and a thick pellet. So during PCR, once the magnetic beads and PCR were in the centrifuge tube, they did not mix. But once placed on the magnetic beads, the beads went towards the magnetic and a back pellet was formed with a green supernatant. And then after PCR, the volumes in each tubes were equal. The supernatant was clear, while the pellet was brown when purifying through PCR. These are our nanodrop results, our concentrations, and also our 260/280 ratios. This is also our gel electrophoresis results. This only shows a positive band from this side. This is our fluorometry quantification results, and these are our sequencing results. So for our sequencing results, we had for sample 333, our number of reads were 14,406. For sample 342, there was 17,582 reads. For sample 343, there were 16,275 reads. And for sample 344, there were 14,862 reads. So at the phylum level, it showed that the Bacteroidetes were the most present organism for sample 333, while for sample 342, 343, and 344, the Firmicutes were the most present organisms in those samples. At the order level, Bacteroidetes was the most present organisms in sample 333, while for sample 342, 343, and 344, the Clostridiales was the most present organisms. So at the genus level, that's where it kind of gets a little switched up a little bit. Sample 333, the most present organism was the Bacteroides, while for sample 342 and 343, the most present organism was the Rhamnococcus organism, while for sample 344, the most present organism was the Clostridium. So in these classifications, there were names such as other and unclassified at this type of level in the top 5 of all of these samples, and it just means that there were bacteria. It could have meant that there were new bacteria that hasn't been discovered yet, or just some other contamination that, as you go more specific down the classification, it would have been revealed more. So, referring back to our nanodrop results, the extraction of the DNA was successful because we had enough DNA. As you can see, we had enough. However, for sample 2, there was impurity within the 260/280 ratio. It had a ratio of 3.56, which kind of tells us that there was a high RNA contamination in that sample. But for other samples, everything else was pure and there was enough concentration. When using the gel electrophoresis, the reaction did not work well as expected for the samples. However, the positive control reaction did work well because the band showed, as shown in this picture right here. Since our samples did not have a band, it makes us less confident on the accuracy of our results. The quantification results for the index PCR/cleanup showed that only our positive control had enough concentration for sequencing. As you can see right here, we didn't have enough for any of our samples, even the negative control, but that's a good thing because you don't want any concentrations to show up for the negative control. So the number of reads in the positive control was 10,313. At the phylum level, the most organism present was the Proteobacteria at 98.93%. At the genus level, the most organism present is the Escherichia at 78.49%. These results tell me that as it goes down the classification system, the organisms become more specific, and other organisms are now shown. As stated previously, sample 333 had 14,406 reads. Sample 342 had 17,582 reads. Sample 343 had 16,275 reads, and sample 344 has 17,862 reads. These numbers are adequate to base my analysis because there are more than 1,000 reads. In the phylum level, sample 333 had 64.1% of the Bacteroidetes, while sample 342 had 98.21% of the Firmicutes. At the order level, back to the phylum level, for sample 342, it only had 0.13% of the Bacteroidetes, while for sample 333, it only had 28% for the Firmicutes, and it kind of shows that they are different in the number of organisms present at their phylum level, and it's kind of consistent throughout the order and genus levels too. For sample 333, 64% was present in the Bacteroidetes, while 0% was shown in sample 342. While for 342, the highest was the Clostridiales, while at 92.6%, while 24.2% was of the Clostridialis was present in sample 333. It goes again to show that one is more present than in the other sample. And at the genus level, the highest was the Bacteroides for sample 333, while sample 342 had 0% of that organism present. And for 342, the highest was the Vermiculus, while for sample 333, it only had 5% of that organism present. So it just goes to show that they weren't different and that they do have different gut microbiomes. And that kind of supports the hypothesis that we did state that if the dogs that consumed more processed food, which was 333, it will have a different gut microbiome than a dog who has a more vegetarian diet, which was sample 342. And research has also shown that the ratio of if you have more firmicutes than Bacterialis means that there is obesity and in that animal. So sample 342, since it had a high percentage of firmicutes and a low percentage of, or 0% of Bacterialis, it kind of shows that there might be some type of obesity or weight gain in that animal rather than in sample 333. And that concludes my presentation. Thank you.